Let's start by understanding what antibodies, or immunoglobulins, really are. These Y-shaped glycoproteins play a crucial role in our immune system. Their tips are where they bind to invaders, which is key to how they protect us. So antibodies aren't just molecules, they're active defenders, recognizing and neutralizing threats in our bodies. So what exactly are antibodies? They're glycoproteins produced by B lymphocytes, mainly found in serum and tissue fluids. The key thing to remember is their antigen specificity. They can target specific invaders. They're major plasma proteins forming the first line of defense in our immune system. This interconnected role makes antibodies essential for immune protection. Now, let's dive into the antibody's molecular structure. It's made of two heavy and two light chains linked by disulfide bonds, creating that classic Y shape. The variable regions at the tips are where antigens bind, giving specificity. The hinge region adds flexibility, and carbohydrates attach to the constant region. Enzymes like papain and pepsin can cleave antibodies into FAB and FC fragments, which is important for their function and study. Understanding how enzymes cleave antibodies helps us see their functional parts. Pepsin cuts IgG into an FAB'2 fragment, keeping both antigen binding arms connected, while papain splits it into two FAB fragments and one FC fragment. This breakdown reveals how antibodies can be dissected into pieces that retain specific functions, which is crucial for research and therapeutic uses. Let's explore antibody structure and function together. Heavy chains define the immunoglobulin class, like IgG or IgA. The constant region is conserved, while the variable region forms the antigen binding site. After digestion, FAB fragments keep binding ability, and FC fragments contribute to biological activity. Light chains come in kappa and lambda types. This integration of structure and function is what makes antibodies so versatile in immune defense. Comparing immunoglobulin classes shows us their diversity. For example, IgG has 5 subclasses and a high serum concentration, making it the most abundant. IgM is the largest with strong complement fixation, while IgA is important in secretions but doesn't fix complement. IgD and IgE have lower serum levels and different functions. These differences highlight how each class plays unique roles in immunity. Continuing with immunoglobulin comparisons, serum half-life varies widely. IgG lasts about 23 days, much longer than IgE's 2 days. Only IgG crosses the placenta, providing fetal immunity. IgE triggers mast cell degranulation, key in allergic reactions, while IgG, IgA, and IgM contribute to bacterial lysis and antiviral activity. These functional nuances explain how antibodies adapt to different immune challenges. Looking at molecular structures, IgG is the classic Y-shaped monomer. IgE and IgD are also monomers but have unique features. IgM stands out as a pentamer, with 5 units linked, making it large and effective in early immune responses. IgA often forms dimers in secretions with a secretory component, protecting mucosal surfaces. This structural variety supports their specialized immune roles. IgM is our body's first responder among antibodies. As a pentamer, it has 10 binding sites, making it excellent at agglutination. It constitutes about 5 to 8% of serum immunoglobulins and is the main antibody during the primary immune response. IgM also activates the classical complement pathway efficiently, providing strong early defense against infections. IgA mainly protects mucosal surfaces. It exists as a dimer in secretions like saliva and tears, with an added secretory component for stability. IgA prevents microbes from sticking to surfaces, promotes phagocytosis, and activates the alternative complement pathway. Its presence in breast milk and mucus highlights its vital role in oral and mucosal immunity, acting as a frontline defender in our body's exposed areas. IgD is less abundant but important. It's a monomer found mainly on B-cell surfaces, acting as a receptor. Though present in trace amounts in serum, its key role is signaling B cells to produce antibodies upon antigen detection, so IgD is crucial for initiating immune responses, even if it's not as prominent in circulation. IgE is the antibody behind allergic reactions. With the lowest serum concentration, it triggers type 1 hypersensitivity responses. Beyond allergies, IgE helps defend against parasitic worms, playing a specialized role in our immune system. Its monomeric structure supports these unique functions, distinguishing it from other antibody classes. Finally, let's summarize the key functions of antibodies. They prevent microbial adhesion, especially secretory IgA, on mucosal surfaces. They neutralize pathogens by blocking their binding to host cells. Opsonization coats invaders to enhance phagocytosis. Complement activation triggers immune cascades, leading to pathogen destruction. And ADCC targets infected or abnormal cells for elimination. Together, these functions showcase the multifaceted defense antibodies provide. Isotypes refer to the different classes of antibodies, each distinguished by the specific constant region of their heavy chains. Allotypes of antibodies are variations in the amino acid sequences of antibody molecules within the same species, primarily in the constant regions of their heavy and light chains. Idiotypes are unique antigenic determinants found on antibody molecules, specifically within their variable regions.